Good afternoon. My name is Dr. Chen Duksin, and I'm the Executive Medical Director at AL102 Pharmaceuticals. Today, I would like to present to you AL102, which is an oral gamma-secretase inhibitor, which we are developing for the treatment of desmoid tumors. Of course, I'll be presenting on behalf of all of my colleagues at AL102 Pharmaceuticals. And to start off, I'd like to thank the DTRF for giving us this opportunity to present our development program to you. So first of all, to present our company, AL102 is a clinical stage biopharma, and it's based on two gamma-secretase inhibitors that we in-licensed from BMS. The first is AL101. It's an intravenous drug, and it's being developed currently in phase 2 for adenoid cystic carcinoma and for triple-negative breast cancer. In both cases, we are selecting patients for this trial based on next-generation sequencing to select patients with Notch-activating mutations. AL102, our sister molecule, is an orally available gamma-secretase inhibitors, and our current plan is to develop it for desmoid tumors, as I'll detail further in this presentation. In addition, we have a partnership with Novartis to develop AL102 for the treatment of multiple myeloma, together with anti-BCMA therapy. So gamma-secretase inhibitors work on the Notch pathway. The Notch pathway, as you are well aware, is a highly conserved biological pathway that controls the differentiation and maintenance of stem cells. It works through 5 different ligands that connect to 4 different extracellular receptors, Notch 1 through 4. Then the activation goes through a cleavage step with gamma-secretase, which in turn releases the Notch intracellular domain, which then goes to the nucleus and interacts with genes there. In certain cancers, this pathway is dysregulated, and you have activation without need for ligand connection. You have this overexpression, over-release of NICD into the nucleus, which can lead to further proliferation and mutations, metastasis, and resistance of these tumors. AL101 and AL102 both work by inhibiting gamma-secretase and basically blocking this cleavage step, therefore inactivating the whole signaling process of Notch. To connect this to desmoid tumors, so as you know, beta-catenin pathway is involved in desmoid tumors, in which 90% of patients have a mutation in the beta-catenin itself, leading to accumulation, while the other 10% with familial polyposis have a mutation in the APC that also leads to the same accumulation. It has been shown in clinical studies that there's a crosstalk existing between the Notch pathway and this beta-catenin pathway. And it has been hypothesized that this crosstalk is mediated by HES1, which is a downstream gene that is activated by signaling of the Notch pathway. I'll go back to this HES1 molecule later in the presentation. Okay, so AL101 and AL102, as we said, small molecule gamma-secretase inhibitors. These two sister molecules basically share the same chemical structure and backbone, with some differences that you can see highlighted in the red circles. Both of them have long half-lives. AL102, its structure was designed for oral administration. So in an in vitro cell-based assay, we can see that both drugs, and specifically AL102, are strong inhibitors of the Notch pathway, shown by low nanomolar concentrations of inhibition across the 4 different Notch receptors. And this is in comparison with other gamma-secretase inhibitors that are in development, namely nirogacestat, developed by Pfizer and out of SpringWorks, and also gamma-secretase inhibitors developed by Roche and Merck. On the bottom, we can see in vivo human tumor xenografts grown in mice, and we can see anti-tumor activity in various types of cancer, including breast carcinoma, pancreatic carcinoma, and lung cancer. Okay, so after completing the full battery of toxicological studies, AL102 went into phase 1 clinical development, and in a phase 1 ascending dose study, recruited patients with advanced solid tumors. The study was split up into 2 arms. Arm A tested daily dosing of 24 patients in ascending doses, starting from 0.3 milligrams a day and up to 2 milligrams. Arm B tested an intermittent scheduling dose, basically of 2 days on and 5 days off, starting with 2 milligrams a day and then ascending to 4 milligrams and then to 8 milligrams. With regards to safety, so in the daily dosing, the 1.5 milligram daily dose was found to be the maximal tolerated dose, with 1 dose limiting toxicity in 6 evaluable patients. In the intermittent dosing arm, the 4 milligram dose was found to be well tolerated as well. The safety profile mainly constituted of the expected GI side effects of gamma-secretase inhibitors, specifically diarrhea and nausea. With regards to efficacy, so we'll talk about both target engagement and pharmacodynamic measures and some clinical data that we've shown for our molecules. So first of all, the pharmacodynamics. So let me walk you through this slide. We see on the left side daily dosing and on the right side intermittent dosing. So first to the daily dosing, the y-axis shows the relevant inhibition of HES1, the gene that we mentioned before involved in the crosstalk with beta-catenin. We can see at the steady state that after the daily dose, from doses ranging from 1.2 milligrams and above, we see continuous inhibition of at least 70% in the expression of HES1. On the right side, in the intermittent dosing, again at steady state, so after the 2 doses on day 1 and day 2, we see inhibition of up to 80%, and this lasts for at least 4 days out of the week. This data gives the basis to use this intermittent dosing in our clinical trials, as we've been able to show both tolerability and target engagement. Some clinical data. So gamma-secretase inhibitors are already known to have some effect for desmoid tumors. So this has been shown also with our drugs. With AL102, one of the patients in the phase 1 trial had desmoid tumors, and this patient achieved stable disease with shrinkage of above 16% after 9 months of treatment. And this is without this patient having been shown disease progression before entering the trial. With the sister molecule of AL101, 3 patients were included in the phase 1 trial. 2 of them had shown confirmed partial responses, and 1 of them was stable disease. The 2 patients with partial responses continued treatment as part of an expanded access protocol and maintained their responses. An additional patient was added to this expanded access protocol and showed a dramatic response that we'll show it in the next slide, and is still continuing treatment to this day. So we can see here on the left, we can see a sizable tumor in the abdominal cavity in both panels on the right before treatment. After treatment, we see sizable and marked reduction in this tumor that is very evident after treatment with AL101. So to summarize, AL102 is a highly potent, selective, orally available gamma-secretase inhibitor. We've been able to show robust tumor inhibition in various tumor xenografts. Phase 1 study showed that oral dosing is safe and tolerable, and this is both in daily dosing and in intermittent 2 days on, 5 days off dosing. We've shown in both of these schedules continuous inhibition of NOT signaling. And also some patients, including in our clinical program, have shown encouraging clinical activity of our drugs in desmoid patients specifically. So all of this is leading us to initiate a clinical trial of AL102 in desmoid tumors in the first half of 2021, and we look forward to collaborating with all of you on this study. With that, I'd like to thank you, and we look forward to answering your questions in the live session in September 25th.